In this lesson, we'll be looking at the neurotrophic and inflammation hypotheses of depression. To get us started, we are going to look first at the HPA axis because that is our central system that is involved in responding to stress. And what happens, of course, is once we experience stress, it activates certain parts of our body, again, primarily the HPA axis with the hypothalamus, pituitary, and adrenal glands, and all of those kind of work in conjunction with one another and keep each other activated as long as the body is exposed to stress. So what does this have to do with depression? Well, when the HPA axis is activated, certain things happen. So cortisol is released, and we know that when we see an increase in glucocorticoids, cortisol, that certain things happen in the brain. And the things that we see that are related to depression is we do see a decrease in prefrontal cortex activity. When the body is handling stress, it's going to send all of its resources to those subcortical survival modes of the brain. The prefrontal cortex is not a survival component of the brain. We don't need higher order cognitive processing to handle fight or flight responses. So our prefrontal cortex activity tends to decrease. Remember, prefrontal cortex activity, we want that to help keep the subcortical areas in check. So if the prefrontal cortex activity is decreased, that means that hyperactivity that's already being experienced in those subcortical areas because of the stress is not being kept in check. So that's going to be one potential negative outcome of experiencing stress. We also see decreases in hippocampal functioning. The hippocampus, it is a primary place where there are glucocorticoid receptors as well as BDNF receptors. We're going to come back to brain-derived neurotrophic factor shortly, which is the BDNF. We're going to focus right now on the fact that that is where a significant amount of cortisol is produced and signaled in the hippocampus. So because we know that there's a significant amount of cortisol produced in the hippocampus and there's lots of receptors there for glucocorticoids, that is where a lot of activity is happening when someone is stressed. And the hippocampus, very much involved in memory. One of the things that we see happening when someone is exposed to chronic stress is the way that they're able to function with their memory changes. We typically have memory that relies on external cues for learning, for adapting. We respond to our external sources. However, when we go into survival or fight or flight mode, that tends to be minimized and we revert to what's called a habitual learning mode. And this is because the hippocampus is overwhelmed with cortisol, so it can't function appropriately. It can't function fully. It's got to kind of cut down its resources and just do the bare minimum. And when we go to more of a habitual learning style, we tend to focus on things that we're already familiar with, habitual, and things that are more internal. We don't look to our external sources because oftentimes if we're experiencing stress, we see the external environment as a threat. So we look more inward for how we're going to cope and respond. And if your subcortical, amygdala, hippocampus is all active because of the stressors, that is going to be what you're using to prime your learning and how you're responding, or those things such as fear and sadness. And so that can create an increase in that depressive responses to the environment or depressive responses in general. Now, I've already kind of referred to the fact that we see a lot of increased subcortical activity when we're stressed because of the glucocorticoid increase. That comes back to there being that increased amygdala activity. So again, when you combine all of this increased amygdala activity with decreased prefrontal cortex and decreased hippocampal functioning, you find yourself in a place where you are relying heavily on depressive factors, and you're highlighting the features of your brain which are going to focus on sadness, fear, depressive type behaviors, feelings, and symptoms. Now, one thing to keep in mind is going to be the brain-derived neurotrophic factor, and this is what I wanted to highlight on this slide. So for short, that's BDNF, and putting this back into the system of the hyperactive HPA system, as well as increased glucocorticoids, what happens is we also see a decrease in BDNF. Now, the reason for that is the hippocampus is a primary site where BDNF is produced. However, if the glucocorticoids are saturating the hippocampus and making it its its primary function is to handle the glucocorticoids that have come about because of stress, BDNF can't be produced, or it's not being produced as regularly or as efficiently. So what we see is that there's actually a decrease in BDNF in the hippocampus when we see increased stress. And because of the decreased amount of BDNF, or the factor that is helpful for neurogenesis, we see a decrease in hippocampal volume. So one of the things that we see with depression is that individuals who have depression have decreased volumes of their hippocampus. We're not quite sure if that is a symptom of depression, or if someone who has a decreased amount of hippocampal volume might be a risk factor for depression. We'll definitely talk about that in more extent a little bit later, but the idea here is that, again, you're just reducing neurogenesis in the brain, which puts the brain at more risk for experiencing depression because it's not able to generate those new fresh cells like we would expect to see. To further highlight the role that BDNF potentially plays in contributing to depression, there are two studies I want to talk about. In the first one, they have found that individuals with major depressive disorder have reduced levels of BDNF serum in their system. And then another study that's been done, they actually knocked out BDNF in rats in their hippocampus. And when they did that, that actually produced depressive-like symptoms in those rats. And the way that's measured in animals is usually by looking at whether they have sex drive, whether they're interested in food, whether they kind of have some lethargic behaviors, and they did find those behaviors in rats after the BDNF was knocked out. So this role of reduced neurogenesis does seemingly speak to a contribution to depression, perhaps because there is limited amount of neurogenesis occurring and helping with some of that cell growth and to helping prune away the cells that are no longer helpful through the neurogenesis process, which is not happening as frequently when individuals are experiencing a lot of stress. And now this brings us to the role that inflammation plays. So inflammation, this first came to the attention of researchers when they realized that there was a significant proportion of individuals who had autoimmune disorders who also had depression. That's a very highly comorbid combination with autoimmune disorders and depression. And so they wanted to look into that. And what they did find is that there is this bidirectional relationship such that individuals who were in a more pro-inflamed state show increased depressive symptoms and stress. And then because they show the increased stress, that contributes to continuing to experience a pro-inflamed state as opposed to an anti-inflammatory state. So it's a very bidirectional relationship between inflammation due to stress as well as how the inflammation then can increase stress at the same time. So it's, again, kind of a vicious cycle in a way. However, interestingly, when the body experiences an inflamed state, it goes into what we call a sickness role. And the reason for that is when your body has an inflammation, it takes a lot of metabolic energy to address that and to heal that. And as a result, it will kind of shut your body down so that it can focus on healing. Again, it's going to onset these symptoms such as fatigue and hedonia. And these aren't necessarily going to be the result of depression, but the symptoms that we see when someone is sick, again, when someone is sick, they tend to be tired. They want to rest. They don't have all this energy. They might not be able to sleep as well, or they might sleep too much, kind of hypersomnia in addition to insomnia. And those behaviors are not because of depression necessarily, but because that's the body's evolutionary way of trying to heal itself. It needs to kind of stay home and not go out and try to do as many activities as possible, not engage in higher order thinking, because that takes a lot of energy. So these sickness behaviors begin to mimic depression. Well, if you stay in this constantly upregulated cycle of you have inflammation, so you increase your stress level, you increase your APA axis response, which that then in turn increases your inflammation, you have a hard time getting out of that rhythm, and those sickness behaviors can almost become more permanently into an actual depressive episode, a depressive onset. So in looking at this pattern of how these different steps affect each other, again, when we have pro-inflammation, that's going to upregulate the production of cytokines in the body to kind of help fight that infection, help fight that inflammation. That is going to upregulate the production of glucocorticoids because your HPA axis system has now been sent on high alert. So it's going to increase the production of those glucocorticoids to kind of fight that stress. We already know what happens in the next step. When those increased glucocorticoids happen, we see reduced prefrontal cortex activity, reduced hippocampal functioning, as well as increased amygdala activity. But what we've added on to the model here is, again, this is a vicious cycle. So you continue to increase your inflammation when you have increased glucocorticoids and increased amygdala activity and decreased neurogenesis in the hippocampal functioning, right? You continue to increase your cytokines. So you're hopefully seeing that this is just a vicious cycle with how inflammation in the body reacts with stress, which then just kind of continues to increase the inflammation in the body. But one thing I want to highlight specifically is how it affects serotonin and neurogenesis. So one of the things that we see is when we have an increase in glucocorticoids, that's actually going to affect the production of serotonin. So the dorsal raphe is an area where serotonin is largely produced. It's a primary serotonin generator, and that has a direct connection into the hippocampus. And so when that serotonin is being produced from the dorsal raphe area of the brain, that is going into the hippocampus and it's helping to trigger neurogenesis. However, if we see a reduction in serotonin production because of the increased glucocorticoids, that is also inhibiting neurogenesis. So again, it's all coming together, it's all a vicious cycle, and that is one way that we see the connection between inflammation and depression.